Alright, so today we're going to be talking about viruses and bacteria. So they are not the same thing, however, they can both cause illnesses, and we're going to talk about how they cause those illnesses in just a second. But first we're going to do a little bit of vocabulary, so let's start off and talk about viruses. Now viruses are non-living particles composed of nucleic acids, so just like DNA, they are composed of nucleic acids. However, they are just enclosed in a protein coat and are smaller than the smallest bacteria. So even smaller than the smallest bacteria that you have ever seen about, heard about, they're going to be smaller than that. Viruses are extremely small. So viruses, however, are considered to be non-living. They are not living due to the fact that they do not exhibit all of the criteria for life. So they do not respirate and they cannot reproduce on their own. So some of these things that they need to be able to be considered alive, they do not meet those criteria. Now the only thing that a virus can do is replicate itself. However, it has to have something called a host cell in order to actually be able to do even the replication. It cannot replicate on its own. So a host cell is a cell in which a virus replicates itself. Now a bacteriophage is a little bit different because it's a virus that infects a bacteria. Sometimes it might just be called a phage for short, and we're going to talk about how this exactly occurs. Now Vic number one is that viruses are small, non-living particles that can cause illnesses. So like for instance, the flu is caused by a virus. It's caused by many different types of viruses. Alright, now how are they structured? So first off, we're going to look at the capsid. Now the capsid is the outer protein coat that surrounds the nucleic acids of the virus. So for instance, a virus is contained, its nucleic acids are inside, and the outside, what's keeping all those nucleic acids inside is just a small protein coat. So you have many different proteins containing in all those nucleic acids and keeping them all in one place. Now, the nucleic acids of the virus contain instructions for copying the virus. So basically, the only thing that this virus can do is it can make copies of itself from this host cell. That's all that its DNA instructions tell it how to do, is replicate itself and copy itself. Now, a virus must attach to a host cell in order to replicate itself. So it has to attach to a host cell before it can ever replicate, before it can ever make copies of itself, and before it can ever become truly deadly in terms of how strong a virus can possibly be. Now, in order to attach itself, one of its proteins, so again, remember, nucleic acids got the protein coat over it, one of its proteins must interlock kind of like a puzzle piece with a receptor site on the host cell's plasma membrane. So remember, the plasma membrane is what is keeping all of those organelles in place in that cell. So what is going to happen, the virus is going to be in the bloodstream, it's going to come along, or it's going to be wherever it is in your body, it's going to come along, and one of the proteins is going to match with this cell's receptor site. So kind of like two interlocking puzzle pieces. Think of the virus as one of those puzzle pieces and the receptor site on the plasma membrane of the cell that it's attaching to as the other puzzle piece. Now each virus has a specifically shaped attachment protein. So just like a puzzle piece will only fit with another puzzle piece or one other puzzle piece, same thing with a virus. Has a specifically shaped attachment protein. So it can only fit with a few cells. It might be able to fit with maybe 2, 3, 4, but it's never going to be able to fit with something like a million different types of cells. It's only going to be able to fit with a certain finite number of cells. And some viruses, like smallpox, are actually species specific. So whereas things like the flu, it can travel between species, it can go from humans to animals, smallpox can only be found in animals. Now because of that reason, smallpox was actually eradicated. So we do not have smallpox anymore. But as you're familiar with, every single year we have a new strain of the flu. That's because it can jump back and forth. It makes it really hard to create a vaccine that would be able to eradicate the flu. So how do viruses replicate? How do they make copies of themselves? So once a virus attaches to a host cell, the virus enters the cell and takes over its metabolism. So all of those processes that are occurring inside the cell, the virus will take over all of those processes. The virus will take over the cell's genetic material, so its DNA, its organelles, anything inside that cell will be taken over by the virus and use its materials to make copies of itself. So in essence, it's kind of like it's breaking into a bank or something and stealing all of its money and using all that money to buy something that it wants. So it's using resources. It is basically confiscating those resources, taking those resources from the protein, and then, or I'm sorry, from the cell, and then going and using those materials and those processes for itself in order to replicate itself, in order to perform its main function. Now eventually, what's going to happen is that the host cell is going to become too large. There's too much of that virus inside the cell that has replicated itself, and it will burst open. This allows the replicated virus to infect other cells. So whenever it bursts open that cell that it has attached itself to, once that happens, all the viruses that were inside that cell, causing it to come apart, causing it to burst open, that's going to go and they're going to infect other cells. Now, a provirus is a little bit different. This is viral DNA that has actually become integrated into the host cell's chromosome. So it has not just broke into the cell in order to use it, in order to use its genetic material to make copies of itself. A provirus is actually going to integrate itself into the host cell's chromosomes. So this may not affect the functioning of a cell, so it may not cause the cell to burst, it may not cause the cell to stop using all of its genetic material. However, but every time that cell replicates, so every time that cell goes through the process of mitosis, the provirus is going to be replicated with it. So you have the option, or you have the problem that these cells might be replicating and creating their own DNA and creating with them this provirus. So example would be the herpes 1 virus, which causes cold sores. So basically the provirus might not always be shown, but it is always there. So it is always replicated with that cell. So that's why people will sometimes get cold sores on their lips, but they don't always have a cold sore. It's caused from that herpes 1 virus. Now a retrovirus is another type of virus. This is going to be an RNA virus with the most complex replication cycle. These viruses do not contain DNA, they only contain RNA, and we're getting ready to see why. So once inside a cell, the retrovirus is going to use the enzyme reverse transcriptase, and that's going to create the DNA that it needs from the RNA, which will become integrated, so kind of going into that host cell, integrated, become one with that host cell's chromosome, and become a provirus. So an example of a retrovirus would be something like HIV. So again, you may not always see symptoms of HIV whenever you first get it, but that provirus, as it continues to replicate in the cells, as they continue to divide, it's just going to cause more and more and more of a problem. So HIV, or human immunodeficiency virus, is going to infect your white blood cells. Whenever those white blood cells continue to replicate, they're continuing to create that provirus and continuing to spread them throughout the body. The retrovirus can infect other white blood cells whenever those blood cells that are already infected burst, just like a regular virus. The destruction of white blood cells interferes with a person's ability to fight infection. So HIV, although the common misconception is that you will die from HIV, you will not die from HIV. What you will do is you will die from a disease or an infection like the flu or something after you have HIV, because your body is unable to fight off that infection, to fight off that virus. Now, two more different types of viruses. We have prions, which are a type of virus composed of proteins, but they have no nucleic acid to carry genetic information. So how these can be detrimental is that basically they will cause the improper folding of a protein, the improper structure of a protein, or it causes a protein to form an improper structure. This can happen in the brain, and if it does, our example, we can get mad cow disease. So cows, if proteins in their brain develop inefficiently or incorrectly, they can have mad cow disease. And a viroid, the last type of virus we're going to talk about, is composed of a single circular strand of RNA with no protein coat. And this is more so seen in plants. Right, now bacteria. For bacteria, the entire assembly of bacteria is going to be referred to as prokaryotes. So they are single-celled organisms. There are two kingdoms of bacteria, archaebacteria and eubacteria. So archaebacteria is going to be the first one that we talk about, and they live in very extreme environments. Often there's not even oxygen present where the archaebacteria live. They are autotrophic and create their own food, which autotrophic means that they create their own food from some type of environment. Some of them can even release things like methane and contribute to greenhouse gases present in the atmosphere. Again, that's going to be the archaebacteria. Now, eubacteria, however, live in places that are more hospitable than the archaebacteria. So these are the ones that might live on your kitchen counters if you don't clean them closely enough, things like that. Some eubacteria are parasites, so they obtain their nutrients from living organisms. So this is going to be what would cause you to have an infection or something bacterial, some type of infection or illness. Now, some are photosynthetic autotrophs, which just means that they create their own food from the sun and from the process of photosynthesis. Some are chemosynthetic autotrophs, and what they do is they break down inorganic compounds made of nitrogen and sulfur and release the energy of the bond, which is called chemosynthesis. So they break down those bonds, they break down that chemical compound, release that energy, and that's how they get their energy. So this is our drawing of our bacteria. This is what most of them will look like. We'll go from the inside out. So in the very middle, enclosed in your plasma membrane, cell wall, and capsule, you're going to have your chromosome. And normally, this is one large circle or one large circular chromosome, not like our DNA at all. So one large circular chromosome that has all of the different types of genetic information that this bacteria will need to live and need to thrive and eventually be able to replicate itself. Now, outside of that, you have your plasma membrane. Similar to how we have our cell, you're going to have your plasma membrane kind of enclosing all that in and keeping ribosomes inside the plasma membrane as well. Outside the plasma membrane, you're going to have a cell wall. So similar to what a plant cell has, this is going to be protecting the plasma membrane as well as protecting the chromosomes. And then finally, outside of your cell wall, you have what's called the capsule. The capsule is kind of like an encasing where it's going to be protecting just another layer of protection for that bacteria. And around the capsule, bacteria do not have legs or any kind of limbs like we do, so they have to rely on things called pili. And these are little, basically almost like hair-like structures that can make this bacteria be able to move by way of locomotion. So it's just swimming around in its little liquid medium or whatever it's moving in by way of these pili. Also, the flagellum are going to be more so of a more powerful motoring agent. They're going to make it to where it can move a lot quicker and a lot more efficiently. And those are going to be longer looking little hair-like structures. Now, how do bacteria reproduce? So bacteria reproduce asexually in a process known as binary fission. So how this works is that bacteria are going to make copies of its chromosomes, and those copies of its chromosomes are going to attach to the cell wall of the bacteria. Once that happens, the cell is going to continuously grow larger and larger, and finally that's going to cause the chromosomes to move to the opposite ends of the cell, opposite ends of that bacterial cell. Eventually, similar to what we have happening in the process of mitosis, a partition is going to form between the cells and separate the cell into two different cells. They are going to be very similar and possibly even identical to each other because this is just a copy of that bacterium's chromosome. Some bacteria, though, reproduce sexually through a process called conjugation, in which one bacterium will transfer all or part of its chromosomes to another bacteria through a bridge-like structure called the pilus, which is, in plural, the pili, like we were looking at with our last board. Now, bacteria, they can harm people. We are familiar with bacteria in our everyday lives. They can harm people in two ways. First, you can have a massive growth of bacteria can interfere with normal function of your body tissue. So, for instance, gangrene. Gangrene can cause your body tissue to start to decay. And lastly, they can release a toxin that can directly attack the host. So a toxin is any kind of poisonous substance that if you ingest it or if it's present in your body can cause you severe harm.